Okay, as you can see, without telling you, the title was changed a little bit to be a little bit more specific, but as you'll see, it's really a principle that I'm trying to share rather than something very rather specific. So today I'll be telling you about hypothyroidism genetics, how can genetics can help understand this fairly complex disease, and more importantly, I would like to engage into the functional insight from gene-based analysis. And this, I hope some of you had the chance of seeing the poster, but if not, I'm sure I'll be able to cover it, most of it. So my talk will be divided to very short section. The first one that I'll start with will be really to understand a little bit more about the disease. And I think as a message, I would say that first learn the physiology so that it can make sense what you find. I mean, without this, I think we are just data scientists that crunching numbers. So for me, it's very important coming from the biology, the more medical thinking and so on. So there are a few messages that I'm trying to convey throughout this talk. And one thing, whenever you see this sign, means that it's worth remembering because it's a more important note. It doesn't say that the other stuff is not important, but they just say that please note to this one. So let's start with the many phases of a single disease. So what's hypothyroidism? It's a disease that the name is built from two parts. The hypo means too little, very little. And thyroidism, it's the thyroid hormones that are not producing as they should. And what you see here in the scheme are the how they look like. And basically, from this, you can understand that we are talking about lack of thyroid hormones. And for short, just for simplicity, I'll call this disease hypo-T, just for abbreviation. So while it seems very simple, I hope to convince you that this simple definition, it's quite complex disease with many causes. And when I talk about the thyroid themselves, the hormone, what it is good for. I mean, as I said, let's understand a little bit of their biology. So thyroid hormones are really essential for normal development of many human tissues in our body on the development side. However, thyroid hormones also regulate our metabolism virtually for all cells and all organs in the human body. And quite importantly, the thyroid hormone are needed throughout our life, as opposed, by the way, to other hormones like growth hormone that they are needed at a certain time of our life. So if everything is good, nothing will happen in the long run. So this is not the case. And indeed, as I mentioned, the effects are on many cells and tissue and organs like mitochondria, fibroblasts, and so on, and on basically all organs that needs a metabolism, which is all organ in our body. So it's very general in a sense. So what happens when it doesn't work well? So I think many of us can see that they have many of these symptoms, like fatigue, like foggy head, foggy brain after 3 days sitting in rooms, like and muscle cramp, and sometimes resistant to stand for cold, just and weight gain and so on. So the symptom, you see that they are quite general. It's not like you have one symptom you can say, okay, this is the disease. It's kind of low metabolism with a lot of outcomes. So what are those hormones? This is the T4, T3. Those are the main two hormones. And actually, T4 is converted to T3, which is the only metabolically active and functional unit. So this is kind of the precursor. However, in the clinic, we tend to measure this because it's much more prevalent. So just a few numbers. The T4 is 1,000 more than the T3 in terms of amount, concentration in the blood. All T4 are produced in the thyroid, but only 20% are produced in the thyroid for the T3, which means that other tissues are involved in this conversion. the thyroid stores about 3 months of storage of the hormones, which is also important, especially for the clinical point of view when you measure what you measure. And 99% of the hormones are bound, so they are not relevant. So we are talking about really 1% of T3 that is the active component. So I'm mentioning it just because it means that our measurements are a little bit indirect to what is going on for real, okay, because of these numbers. So how we control those hormones in general? So this is a very small negative regulator, negative feedback loop in which I won't go into details, so don't be too afraid. So we are talking about a hypothalamus that produce its hormones to the pituitary gland that affect the thyroid, that stimulate the thyroid gland. But more importantly, once you have the hormones, there are feedback loop negatively to shut down the pituitary and the hypothalamus. So you see that it's a very balanced type of loop. And when you have the effect happen on the thyroid itself, it means primary disease, secondary disease, and tertiary disease, okay? So by the level of this control. And one important point, and I want to mention it now, because this is what I explain now, but what I want to emphasize is that the difference between the disease of hypothy and hyperthyroidism is very small. It's just the balance of these loops that I just discussed. So you can even shift it from this level to this level quite easily. So it's important to note that once you have this disease, it's mainly or often called Hashimoto disease when you have hypothy, and when you have hypothy, it's called Graves disease. So just to see that sometimes even the names are not two different diseases, but different side of the same coin. Okay, so that's an important message to mention. So to summarize this part, we're having primary, secondary, and tertiary, but 95% of the cases are this primary one. Okay, so just to know from numbering. So how we measure them? So the ICD-10 is E03, and you do blood tests, imaging, and more often measuring antibody in the blood. So the first part is really the result of this loop feedback loop that I showed you. The second part is very important because there are in our body antibodies, autoantibodies that attack the gland. And that by itself lead to hypothyroidism. And this is exactly what is defined as Hashimoto disease. Okay, the immune aspect of attacking. And usually the disease is defined by the combination of the two measurements. So now that we know everything about thyroid, let's just go to numbers. In the world, quite amazingly, there are 2 million, in the US, there are 20 millions. Among them, 14 millions have Hashimoto, the same one that I explained. 60% of them are not aware of their disease because, we are all fatigued, we are all, gaining weight. I mean, it's just regular life, right? So it's not, it's underdiagnosed. That's my point. And there is a difference in age of females and male that we'll touch upon it later. I want to touch the women aspect of the disease because the probability of having this disease in women is 5 to 8 times more than male, which is quite extreme. 1 of 8 women will have this disease throughout their life. And about 25% of women that have fertility issues is because of this. So this is much more prevalent and important from public health issue, of course. Okay, so now that we know this, let's go and understand what the association study can help us with. So my question on this is hypothy genetics at all? I mean, I showed you that, as we know, when you talk about disease, you have to think at least on those modifiable factors. And in this specific case, The modifier factor are the known one, like smoking, alcohol, in France. So I'll skip the alcohol, but the exercise, diet, and so on. But specifically, the thyroidism hypothyroidism is very sensitive to pollution and to damage that comes from environmental quality of air and so on, water and so on. So this is very important. But today I won't touch it at all. What I'm touching upon is the non-modifiable factors, specifically the genetics, the sex, and and age. I'll touch a little bit, but not on ethnicity. But those are you are born with. It's not something that you can just play with. So hypothyroidism, indeed prevalence by age. It's become much more prevalent. So just to say, because I mentioned the environmental aspect, in the world, there are 2 billion people that are suffering from iodine deficiency. Iodine deficiency, I showed you the hormone. They do have, I didn't mention it specifically, a lot of ion. Actually, T4 is 4 ion, T3 is 3 ion, iodine, sorry. And you see the map showing that some big area on the globe are suffering for deficiency and some area for excess or too much of iodine. But in the developed world, we don't have this. It's all controlled. So this is environmental aspect that is not relevant, at least to the data that we analyze. So just remember this point. 50 million are clinically outcomes. So I think to wrap this part, I already showed you that it's a mixture of genetics probably and other causes. And for example, the Hashimoto is 2 to 3% in the US. I don't talk today about thyroid cancer, which has a predisposition component, genetic, but I leave it aside, and the congenital disease in which one was born with. It's 1 to 2,000 live birth, which is, again, it's not that rare. It's rare because it's a rare disease, but it's not that rare. And this we call congenital disease. And so what are those genetic associations? I'm sure that many of you are familiar, but just to the few that maybe do not know that well. So what's the goal? The goal is really to find causal link between genetic variants and the phenotype in general. And it could be a disease, it could be a trait. And why? Because we want to understand the biology, the mechanistic underlying, and we want to use it for prediction, for diagnosis, for everything that we talked the last 2 days about. And how? In general, there are 2 models, either family-based or population-based. And today, I'll talk about population-based. And population-based, this is the classical, Wikipedia slide for GWAS, just showing that you have case and control. Case are disease, and you don't have to look at the slide to understand that those allele that are green are disease allele, let's say, because the proportion between the case and control is very evident. And you do just a simple statistics on each and every variant. That's all. Through the genotyping, which is just the way to measure millions of variants. So it's a very simple statistic. It's work on a case control. And importantly, each variant is analyzed independently. So the statistics must be at the variant level. Otherwise, doesn't count. So the results are, of course, the Manhattan plot that we know of. This is the chromosome from 1 to the end. And whenever you see this pile, like in Manhattan, you know that there is a bunch of variants that are focused in specific location. That's it. However, this, of course, just provides correlation, which is very different than causation. So we want to do more. Just to wrap up this pre-story about genetics, I just want to say that GWAS is there for 15, 18 years. And I'm just showing you the change in the last 4 years. You can see that it's shooting up. I mean, there are so many. There are 50,000 published papers on GWAS. I mean, it's really amazing. However, it's very disappointing in a general statement. Why? Because we cannot explain much of the genetic variation. It's not delivered. So biological knowledge was very in general poor, and it's not poor utility. I mean, what can you do with it? So, and there is another substantial issue. The mapping from variant to functional unit to gene is very unreliable. So, fortunately, in the last few years, we have, and we discussed this, biobank that changed completely the story. And this is why I'm so happy to be in this specific session that are talking about making really something that can be used by the community. Without it, it's really a huge hurdle. So Biobank of the UK, which is the one that we are using, is 500,000 people. There is the veteran one with 1 million people. There is the Japanese with 250,000. There is the Finjin with almost 400,000 people. And they all have their pro and con. I won't get into it, but they are important that there are many of them and not only one. So we really can use this. So I'll show you what we are doing. And from now, it's really to show you the result and there's some insight that I want to share with you. Okay, some ideas. So what I show you here is a GWAS done on 18,000 people as cases and 400 as control, 400,000. I mean, really big numbers. And you got hundreds of variants that might be relevant. But if you go and do many independent studies, you can see what I show you in the bottom, that only 20 genes repeats themselves, 21 variant, not even gene, right? Variants repeat themselves. So just for as a start, we say, okay, let's be very, very, very solid. Let's look at this 21. What are they? And when we did it, already we found that they are at some network, maybe not that strong, but a reasonable network. And individually, each gene, because we are taking the variant and say what is the gene that is related, is already reported that they are related to the hypothy and either congenital or to Hashimoto. So we felt that this list is solid. However, to summarize this part, we have many, many GWAS, but the list is very noisy and unvalidated. So we want to do something else. So for that, we develop a method called PWAS, firm and PWAS, that I'll explain very shortly what it is. So the idea is to take the human variants, which is about 97 million that we have in the biobank, and to ignore 99% of them ahead of start. We do not look at them, not because they are not important, because they do not know how to interpret. So we only look at the coding gene, 20,000 coding genes. And for that, we are doing aggregation to genes. And the idea is simple. Instead of what I just told you about GWAS going from the variant to the phenotype, we are doing something else. We are going from the variant to the protein, understand what happened to the protein function, and from that, going to the phenotype. So it's like hoping, you know, like I have a bridge in the middle. And that's important point. So our method that I'm explaining in a minute can analyze de novo and rare variant the same way as we are doing common variant. Doesn't matter. You'll understand in a minute why. We can operate on any SNP. We are using machine learning in our method, and we are using firm as a variant interface method. So let me explain. What's firm? This is the outline of what I'm going to show you. So we first decide what's the impact of each variant on the protein function, not pathology, no nothing, just biochemistry. Then we want to get a gene effect score by heritability model, by thinking what happened if we are talking about recessive, what happened if we are talking about dominant. So really to do both. And then we have case control. So the effect score is analyzed for case control, and then we do pretty straightforward statistics, like in regular GWAS. Okay, what's firm? Firm was extracting feature from everything that we know about protein biology. We basically download the UniProt resource of humans, only humans, and extracted over 1,000 numerical feature. Many of the feature we engineered. So if you want to ask me how and why, it's also published, but this is an important part of this engineering business. And this is done by Nadav Brandeis and Dan Ofer. Nadav was finishing a PhD, Dan is within a PhD in our lab. So the second part, once you have the feature, you do machine learning, and the machine learning was trained over ClinVar. ClinVar is the clinical resource that we are, coming from the public community collection of clinically labeled variant, and we have this machine learning called FIRM, in which this is the performance of the machine learning, all good. But you should ask me, or I should ask myself, why do we need? We have so many. This is just a list of few of those tools. I mean, those. Everybody's using it. Why another one? And the reason is that we want something that will be human specific. We did not use any evolution, no multiple sequence alignment, nothing. It's only human specific, and that's another point. And the second one is that we seek only biochemistry, so we don't look at something else, and we do it in a probabilistic intuition. Namely, if we got a score of 0.7, it means that it is the likelihood of the protein to retain its original functionality, which is 1. Okay, so we have this numbering, so we can capture a lot of features. I don't want to, to including localization, modification, protein ligand, a lot of biochemistry, and of course some we do not on purpose included, including protein interaction and protein expression and non-coding RNA. Very important aspect that we didn't. So just to show you. So we take a variant, go through the inference, get a score from FIRM, and now we have the data to start with, which is 18,000 gene or 20,000 gene for 500,000 people in the biobank. So that's what we do. And the second part is really just to show you that we do the aggregation from the FIRM, and the aggregation I already mentioned through dominant or recessive aspect. And I think in this scheme, you can understand what's the dominant and the recessive, namely, yes. Yeah, please.
How do you train your biochemical damage? What is...
Yeah. So we take those 11,000 features and we train them only on indeed what seems to be clinically, pathogenic already. So there is a jump in logic between this biochemistry and pathology here. You are right. You touched the point, but we overcome this. Yeah. You are absolutely right. So we had this dominant, and this is the work of Ro'i, who is sitting in the audience here, and Miki and Amos, student in my lab. Just one point to mention, that the GWAS is additive in its nature. 0, 1, 2, everybody is using it. And this is illustrated here. But what PWAS allow is also to get this. Because we are doing aggregation, you can have also effect on different part of the genes. Each of them is not that bad, but together they are pathological. So this is another advantage of PWAS. That's an important part, because we are able to gain this information that is overlooked completely by GWAS. So to summarize this, I'll do it very fast because I don't have the time, but I think I'm okay. So I just want to make it fast. So this is all the problems that we try to solve by PWAS. No, not additive, but dominant and recessive. The interpretation is only on function. The mapping is trivial because we start with the gene already, so we don't need the mapping. Also, statistics is much easier because we don't have millions of variants. We just have 20,000 genes and so on. So we have a lot of disadvantage. And this is just an example. This is a gene. Each gene got a score by FIRM. Some of them increased the risk, some of them decreased the risk, and we can really do some aggregation under this assumption of recessive and dominant. So for each person, we have 2 models, and this is done for 500,000 people. Now we have this. Now we can start working on the biobank. Biobank is really an amazing resource in which we have it was collected through 5 years, 15 years ago. And the age of the people between 40 and 70, now they are 15 years older. So it's even better in the sense of clinical aspect. But they do have everything like EMR, self-report, what they eat in the morning, how they like their cider, what's the temperature of the beer in the morning versus in the evening. Everything is there. So it's an amazing resource, both for lifestyle and for genetic. And so this is what we do. We have the coding gene. We have a lot of covariates that I'm not discussing. And this is the 14,000 people that we are analyzing. And when we did this, we found 77 genes. What you see is the color is the risk. So reddish color is high risk, and blue color, the top one, is lower risk. And you can see that we have those genes, just without going into too much detail. When we take these 77 and ask what are they, to our surprise, and for anyone that did GWAS, including us, we know that for most cases, when we take the list of GWAS and try to get some network, you get nothing. That's the default, and that's fine. That's what we always get. But here we got a really strong, and when we look, what are they? This is antigen presenting T cell, antigen processing, interferon, and so on, very significant network. So we were able to show that we were able, through these PWAS, to find the basis, the biological basis of Hashimoto, the aspect of Hashimoto within the hypertension, the hypothyroid, sorry. And we see that 60% of our gene are related to this small section in chromosome 6 that's responsible to all our autoimmunity, our self to non-self recognition and everything. So this is very attractive, of course. And when you do the classical enrichment story, you find, I won't read for you, but you find viral and graft rejection and thyroid and diabetes and all those asthma, all those immune story is pumping up. So the conclusion, PWAS identified almost exclusively the immune axis of the disease. Okay, so usually, by the way, people are talking about the B cell, this aspect, because of the antibody that I already mentioned, the B cell producing antibody, and we were able to show that the T cell is very strong as well. So this aspect of the immunity is also very active in Hashimoto. And then maybe, because I don't have the time, just to tell me when I'm okay? Okay, good. So we are often, remember in my introduction, I mentioned this women's story. So we wanted to understand whether there is something specific in the female that makes it more susceptible to genetic effects. Okay, so this, what you see here is the prevalence in the biobank, in the FinGene. This is why I like the fact that we have different biobank. We can compare and see, because the definitions sometimes are a little bit different. And then you can see, okay, this is independent defined. Can we see the same stuff? So immediately it's give you a very good validation. And we ask what's going on here. Why this huge difference? What's the basis? So I'm just throwing few solutions that we have no idea. It's ideas. Maybe it's X-linked disease. Maybe the time of onset related to the estrogen level of the women. Maybe it's inflammatory status. Maybe it's pregnancy issue. We don't know, right? But it's a lot of things that might do this big difference. So we do what we do. We do what we did before. We are using now PWAS, but separately for male and female. And when we do this, this is the number of male and the number of female. You know that the difference I already mentioned. And this is the control, as you can see, about half and half. And when we do this, we found indeed that the overlap, what you see here in blue, of course, is male. In red, of course, is female, pink even. And in both is all the population. And you can see that we have 7 genes that appears only in female, only when the analysis was done to female only. So that's quite interesting. What are they? And we can see quite a lot of the signal at the male is pretty minimal. This is the overall numbers. So what we did a permutation test to kind of challenge ourselves, saying each and every gene, is it female, male, or let's randomize it and see if it's still important. And to our surprise, all genes fail the exam. So it means that there are no sex dependent, besides 2 genes that were really consistent. This is 30,000 permutation. They consistently show a signature. And what was the signature? To our amazement, it's 2 genes in the chromosome X. We didn't know. There are chromosome X. One is in innate immune response, and the other is the cause for Graves. Remember Graves from the beginning, the hyper T disease. So it's quite nice. I mean, and the last is more like ideas. You know, where are we heading from here? So what we learned from PWAS, just to summarize, if you lost me in the middle, that there is a strong immunity signal, that no clear sex-dependent story that I can put my hand on. The recessive signal I didn't show you, it was not very prominent, 11%. Most of the genes were elevated risk rather than protective genes. And 9 of these genes were also shown in the FIN gene, which is a very good replication result. So now we said the following. Okay, we have our own toy called PWAS, we are enjoying it. But what about the thousands of GWAS that are around? Maybe we can use them and see something interesting. So we took the open target, which is another excellent resource, and we take this open target and we took the top. It's also scored. So the top scoring, 200, and ask what is the association of these guys. And when we did this, we saw, as you can see in the Venn diagram, that the overlap between our result and GWAS result is really embarrassingly low. So what's going on? Why it's so low? And the reason is that GWAS identified a completely distinct set of genes. What are those genes? We took these genes and we made another network from this gene, and you can see a very strong network. What's this network? So for that, I have to tell you that the thyroid has a very unique structure in which the cells are producing with this set of enzyme the hormones. So this is another illustration of the cell biology of what I showed you. So now, if you look at this network, you see that all those genes are from this network, all of them. So basically, we found congenital hyperthyroidism. So completely different story of this disease by GWAS. And then we say, okay, so what's this disease? This is really a severe disease in which the child is born with already affected and non-developed thyroid. It has mental retardation and intellectual disability. It's a really serious one. And when we do this, and I don't want to go into too many details, but then we did PWAS. PWAS is another type of association studies, which is only relying on transcription. And now you get a different story. What's the story? It's about signaling and trafficking, not development, not producing hormone, but trafficking, signaling. So my point as a summary is that PWAS identified immune association, the Hashimoto. GWAS identified developmental program of the hormone and the synthesis of the hormone. And TWAS identified transcription regulation, trafficking and signaling. And my point, my take-home point is that association studies should not be used as a confirmation for one another. They really can, in some cases like this one, show you different angle of diseases, and you should use it to understand better the biology, not just to confirm something that you are looking for. So this is, so we did also some prediction. You know, this is PRS. We ask whether PRS means, to those that do not know, it's just to take the score of each and every individual and to rank it through the population. So those guys are very low risk and those are high risk. And look at this. The risk is 4 times more in the 10% higher population of risk than the lower one. So 4 times more risk if you have the wrong genetics. So that's pretty cool. So we did also prediction, and this is just prediction on the genetics on the coding variant by GWAS. So this is like a PRS of, prediction, and we were able to show the prediction of the coding GWAS, which is also a version of our GWAS that we do only on coding to understand what's going on. So to summarize, GWAS provide explanatory insight. I hope I convince you. The association studies expose different disease etiology. I hope I convince you. Sex prevalence does not, in this case, implying alternative causes. The prediction genetics risk model is pretty good. And I didn't talk about comorbidity, but that's important because we are looking at what you are looking. I mean, really, whether the disease comes first, come later. I mean, I don't talk about it. And this is what we are doing now. We are adding rare variant, which we didn't have in the story. We are now doing the hypo and the hyperthyroidism. We do validation to different ethnic group, causality testing, and so on. And basically what Hashimoto will infer for cancer treatment of immunotherapy, cancer treatment. This is something that some doctor wants us to do and will do. But I cannot stop without saying something important. What next, not for Hypothy, but for Israel? This is very dark days for Israel, really dark. And probably I'm here somewhere. So no academia without democracy. And I want to emphasize, especially for the young people among us, there are some, yeah, that it's really important to know that fight for your rights, fight for what you believe, do not give up, there is no option. And what seems to be very stable, like Israel, used to look like stable, it's fragile in a way that we couldn't even understand. So keep fighting for your belief and for your identity, for everything. Just don't stop. So this is our lab. Amos, Ro'i, and Mickey were involved in this specific one. This is other project in the lab, and I'll be very happy to take a question. Thank you. And you can ask me about Israel, about everything.
So any question?
Thank you. Hi, I've actually got a lot of thoughts. Maybe I can speak to you afterwards.
Sorry?
Yeah. I've got a lot to talk to you.
Yeah. Please do. I'm here till tomorrow.
So I've been actually researching just privately Hashimoto's for the last 2 years. Yeah, and generally what people go and research and find immediately is often not updated information. Right. So I've just taken notes because there's so many things. So you were saying hypothyroidism is underreported, and the Hashimoto's figures there are most definitely underreported because there are actually medical professionals now are saying there's actually 3 stages of Hashimoto's. The first 2, you don't even pick up any problems with the thyroid hormone levels. They're actually normal. And in the first stage, the person actually has no symptoms. So, there's also that.
Of course. So thank you. First of all, thank you. I'll be more than happy to keep discussing with you. But what my point was not now we are understanding what's the doctors, the real doctors for, we do not. But my point is that some of the diseases, not all of them, do have multiple etiology that are hidden. And those multiple etiologies, cause for reason A, reason B, reason C. Usually they are piled. And what we were able to do is to fragment it back to its original different causes. So it's not really the mapping of the disease that was, maybe I used the wrong term saying, but I wanted to say that the immune aspect of the disease was revealed by PWAS, which was not revealed by the other method. So maybe
Yes.
that's more exact.
Yeah.
How,
No, I mean, I get
Yeah.
that part of it as well. But if you kind of have, the updated, which it's very active field at the moment, and quite contested in some cases. But for example, also TPOs is only in
Right.
80% of Hashimoto's.
Right.
And obviously that's when it's been diagnosed at that
Right.
point. But then also just to say the thing of where you had the risk factors for autoimmune, they're finding that stress and trauma is actually one of the highest risk factors.
Yeah.
And when you're talking about what's the why, do women have it so much more
Yeah.
often than men? Because women, experience trauma often a lot
Differently.
more
Yeah.
and differently.
Yeah, differently.
And so that is a big
Yeah.
factor
So I
to
can
account
just tell you, and also you can discuss it with Roy, that is doing a lot of sex-dependent diseases. So we are looking at this aspect of women-related diseases, and it's extremely interesting. I just mentioned it as a comment that the comorbidity is very important. I mean, what they had before, whether they have depression is, so we can define it as a much more rigorous way. But what I wanted to show you that from the puristic genetic point of view, you not always can explain something that is so evident. So the genetic, basically for women in this case, failed. But in other diseases, it was mind-blowing. So you have to be very careful about genetics. But I fully agree. For example, we ignore completely the interaction. For now that I showed you PWAS, TWAS, we can ask what's the interaction between the genes. Maybe then you can have a really good predictor. So we are going there as well.
Sure, thanks.
Yeah, sure.
Yeah, thanks for this wonderful talk.
Thank
I
you.
have a question actually exactly about interactions and networks.
Yeah.
So you showed the classic G-bars and then your P-bars approach, and you used networks to interpret the results at the end.
Right.
How about using networks in the beginning to aggregate weak signals
I love
and
it.
to discover like sub-networks that are associated with the phenotype? Are you working on this, or have you thought about this, or do you think the main value is
First of all, thank you for this
interpretation?
comment, because I was sitting in one, this is the reason to come to meeting. I was sitting in one of the room. I cannot remember who was the speaker. Sorry for, I hope it's not one of you. But I was sitting and saying to myself, there was a talk about the knowledge graph and all this. I mean, really interesting. And I thought, why we cannot take each disease, Roy, this is next for you, each disease, and just show whether we have a network or not, and use this as a proxy for understanding the etiology already from the beginning. So there is this back and forth, top down, bottom up, that needs to be worked out. So it's something that we didn't do. But I must say that those are one of the few cases. We looked at hundreds of diseases. Those are the few cases in which the network were so stable that I could not ignore it. But in other cases like autism, there is a beautiful network of synapses and of chromatin, and it's big, like, and maybe it's 2 stories for autism. But thank you. I think it's a brilliant idea.
Yeah, sorry. Thanks very much for that. That was really, really great. I was just wondering about the kind of the aggregation part of the PWAS as well. Is it like, are you aggregating the variance like heritability models as well? Like, is there kind of, because you've got the 2 heritability models, I suppose, that you're considering?
It's a regression. It's a fairly simple regression, with parameters and so on. But basically, we ask whether we can get, in the regression, something that will be significant if and only if we need 2 mutations in the same or one. That's it, basically. So we kind of put a constraint saying we need at least 2 so that we can refer it to, in this case, by the way, the recessive aspect that I thought will be the highlight was very minimal. But in cancer, we did it for predisposition in cancer. 70% of the story of predisposition is recessive, which I didn't know. We published it recently, I mean, but it was a surprise. So you have to really, you know, it's not one size fits all. That's, we all know. Sure.
More questions? In the meanwhile, I wanted to ask you, because I'm not familiar with this, so how much clinical data are in the
yeah.
data bank? Because typically
Yeah.
when you have
Yeah.
genomic data,
Yeah,
you don't have much clinics.
No.
And you
So
have clinics, you don't have.
I'm telling you, I was working with a lot of biobank. There is nothing compared to this. For example, the FinGene, which is a beautiful biobank, has very little clinical data. Here, it's the opposite. They start by interviewing the people, having the age of diagnosis of each diagnosis, cancer, infection, depression, pain. I mean, really, really deep, including if you ask, it's depend on the application, but if you ask for detail, you have the MRI, the imaging, the bone density, you name it. So it's a very rich and updated, because everyone, yeah, everyone that happened to come back to the hospital, let's say, and it was in the cohort, they have update. So you can play also for, you know, observational data, but see what happened and so on. So it's a very, very phenomenal.
It's amazing.
Amazing, yeah.